Okay guys, welcome to this cell parts flip. The majority of this is going to be about the cytoskeleton, but there was one little part from last time that I did not finish off of when we were talking about mitochondria and chloroplasts. There is this other structure called a peroxisome, and a peroxisome is a specialized metabolic compartment bounded by a single membrane. Okay, remember, metabolic means that reactions are going to be occurring, and it only has one membrane. I gave you a lot of information here because this is really what it does, broken down. It contains enzymes that can remove hydrogen atoms from certain molecules. Remember, it's those hydrogen atoms that make things acidic and eventually could be toxic as well. And then transfers those hydrogen atoms to oxygen, which then makes H2O2, which is hydrogen peroxide. The peroxisome can then take that H2O2 and convert it into water. This all happens within because it has various enzymes that allow it to do this to protect the rest of the cell from these possible harmful compounds. So when would we see this happen? Let's take a look. We would see it happen in the liver. That's the big example I got for you. It's the prominent in the liver to detoxify alcohol and other harmful compounds. So an image of this, check it out right here. Here's our chloroplast that we spoke about. There's a mitochondria that we spoke about, and here is the peroxisome. Okay, again, helps to detoxify, get rid of those harmful compounds, convert it into water so it doesn't hurt the rest of the cell. So that's all I want to say about that. Now, the cytoskeleton is a major component of the cell and often overlooked. Here's why. We know it as a network of rods and filaments in the cytoplasm. And sometimes even in regular biology classes, they're like, oh, we're going to cover these organelles, cytoskeleton, don't worry about it, well, blah. It's just, it gives structure. We're going to go a little bit deeper because the cytoskeleton is incredibly important for a variety of reasons. Two main components of the cytoskeleton you can see here are microtubules, the big thick ones, and microfilaments, which are the thinner rod-like ones. Oh, I put the picture there again. Okay, so functions: cell structure and shape, cell movement, cell division. By cell division, I mean it helps build cell walls and move the chromosomes apart. Oops. Okay, so those are the big three. I thought I had another one in there. Oh, that's later. My bad. Sorry. Cytoskeleton components. So what are we going to be talking about here? We're going to be talking about microtubules. We're going to be talking about microfilaments. And we are going to be talking about intermediate filaments. So the first one we're going to look at is microtubules. Now, I love this table. Okay, it goes through. I can't remember if I stole this from your book. I know it's in your book, but I don't know if it's that table because I stole it from a more recent text that is like yours. Okay, so we're going to focus on these microtubules first. All right, that green stuff that you see for the cell, and what we're going to look at is a tubulin dimer. Okay. Oh my. That's creepy. Sorry. Was that not distracting? Seeing Barbie staring at you. We're going to keep going. Okay, microtubule. I'm sorry, I looked down at the webcam and I was like, that's creepy. Okay, I didn't know she was there. Weird. Like a Chucky version. Okay, so microtubules. Structure: small, hollow tubes made of repeating units of a protein dimer. What's a dimer? Remember, di, two. It is a structure that is made up of two tubulins. One's an alpha tubulin and one's a beta tubulin. That's all you gotta know. Okay? And it's these dimers, though, that's how it grows. It grows from that dimer portion by continually adding tubulin to it. Its size, again, 25 nanometers in diameter with a 15 nanometer lumen. Remember, the lumen is the space in between the membrane. It can be 200 nanometers to 25 micrometers in length. What does this mean? Of all the cytoskeleton, it is the thickest. That's what you've got to know. It's the thickest, okay? And it's what we could say is the largest of them, but sometimes not necessarily in length. Their function is to regulate cell shape. They coordinate direction of cellulose fibers and cell wall formation. So in the cell wall formation, we're going to be looking at bacteria and plants here when we look at cell division further on. Internal cellular movement provides tracks for motor molecules. Now, this is huge. We have talked to this point about vesicles moving throughout the cell and those proteins that are being created, how they know where to go, or the lysosomes moving throughout the cell. How do you think they're moving? They move through the microtubules. They move through these tracks that they provide, and we'll talk specifically how they do that. But the microtubules just aren't there to provide shape. Yeah, they do. They provide shape. They also help with division. But here's the big part. They provide the traffic ways. They provide the roadways throughout the cell. For example, here, we can see here's a vesicle moving along a microtubule. And we'll talk about these little feet right here and how it's ATP powered. Remember, ATP is the source of energy. It's the chemical currency of energy that we all want. And how it moves those vesicles down the microtubule, okay? In order, what it's trying to do is actually get to the axon to be submitted across the synapse, all right? So what you need to know, microtubules provide that framework for vesicles, for motor neurons to travel, not motor neurons, motor proteins, right, for them to travel on, to carry organelles around. So microtubules here, they're made up of centrioles, basal bodies, make up centrioles, basal bodies, and spindle fibers. The other thing that they form is cilia and flagella. So let's take a look at centrioles first. Centrioles, usually one pair per cell. It's located close to the nucleus. They are found in animal cells. 9 sets of triplet microtubules, which I'll show you a picture here in a moment. And they help in cell division. So here's a great pic here. The centrosome is the region where we are going to find the centrioles. It is like the microtubule manufacturing area. And the centrosome here, we have 2 centrioles. Now you can see the 9 sets of triplets. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9 centrioles. Okay? And so that's where we have nothing in the middle. Very important, nothing in the middle. And these are functional in cell division. You might remember from mitosis, where these move to opposite ends of the cell, and then the fibers extend out and grab onto the chromosomes. If you don't worry about it. We're going to get there, okay? Centrioles, though, that's what they look like. That's where they are. The basal body, same structure as a centriole. So 9 sets of those triplet tubulins. And let's take a step back. What's a tubulin? Tubulin is a protein that makes up microtubules. So that's it. It's just a protein. Okay? So let's go back to our basal bodies. Basal bodies have the same structure as a centriole. They anchor cilia and flagella. Hopefully these words sound familiar. So what are we talking about here? All right, take a step back. Flagella are the things that extend off of the cell membrane or the cell wall, right? We're going to talk about those guys in a moment. Looks like a whip-like tail. How it is anchored in. If we look at our TEM here, we can see this section right here this basal body is what anchors that flagella so that the whip just doesn't get lost. So now we're going to take a look at cilia versus flagella. Cilia are short but numerous. Flagella, long but few. Flagellum we only see, usually we see one per cell. Sometimes we see a few, but one is the norm. Function. Their function is to move cells. So some cilia work all together to move cells, or the flagella whips to move the cell, or to sweep materials past the cell. And we're going to take a look at that in a moment. But new evidence is showing that all animal cells, take a step back, not all animals, all vertebrate cells have a singular cilia on each cell. That little cilia that sticks out may function as a signal receiving antenna. And what it does is it detects something from the external environment, conveys that information to the internal environment, and then possibly causes changes in what the cell is doing, its function at the time. Why would this be important? Well, it's huge in looking at development. Okay, and we might talk about it a little bit more later, because this is more, I don't even know if this is mentioned in your book. It might be. Let's take a look, though, at movement. So, yes, this is sperm. Get over it. If we look at the motion of flagella, what we see here is this undulating movement. So, it's how it whips, okay? Like a fish, right? It's a swimmer. If we look at the motion of cilia, though, it's different. You can see this organism here. Looks like a I think it's a protozoan, but it's covered in cilia, right? And how it is moved by the cilia is those cilia work like oars. So think about working on a boat, you're paddling a boat. You have the active stroke where you're pushing through, and then the recovery stroke, and the active stroke and the recovery stroke. So cilia and flagella. Cilia and flagella, their structure is a 9+2 arrangement of microtubules, and it's covered by the cell membrane. Okay, so the cell membrane extends out over that cilia, and flagellum, I should say. It also consists of dynein, which are motor proteins that connect the tubules. So let's take a picture of this. What we see here, 9+2, we see a ring of 9 doublets. Okay, a microtubule doublet, we see 9, and then we see 2 single microtubules in the middle. Now those doublets on the outside are connected by what we call dynein arms with motor proteins. Okay, so usually we say dynein proteins. What's so special about them? Again, here we can see TEM, 9, an image of 9 doublets, 2 singular in the middle. What is so special about those dynein proteins? Well, it's a contractile protein, which means it can shorten and lengthen. It uses ATP. It creates a twisting motion between the microtubules, causing the structure to bend or move. So these guys are literally, they're kind of jokingly called the walking feet, because there's 2 heads to that dynein. And one is going to pick up while the other one stays attached and then move. Yes, this takes ATP. This takes energy for this to occur. All right. And so we can see this, how this walking movement causes the flagella or the cilia to bend and go back and forth. And that's the purpose that we see. And there we also see the pair of dynein arms. And then there's crosslinks between the doublets so that the structure, that ring structure, stays intact as well. Okay, guys, I'm going to shut you off here. We only got through microtubules on the cytoskeleton. So your next flip is going to be over microfilaments and the intermediate filaments. And I'm going to do that one for you right now.